This is the cell cycle M phase. So according to your textbook, there are 6 stages of the M phase. This figure is not from your textbook, but just shows them in a little more condensed form. Interphase is not part of M phase. And otherwise, if we count those up, that means we've got prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis for 6. I want to point out that page 622 to 623 is a nice resource. It's in your textbook. These are some really beautiful images, and they were actually taken by a friend of mine, Julie Kahneman, who's a really exceptional scientist, and she's currently researching the coordination of the mitotic spindle and cytokinesis at Columbia University. So these images are from some of her primary literature publications in Nature, as well as the Journal of Cell Science, and although it might seem like they figured all this out with these cellular events, actually there's a whole lot that is still being worked out, and one of her research goals is to determine drugs which can interact with the cytoskeleton and potentially mediate things like cancer. So certainly you've learned about mitosis many times before in other courses, including in introductory biology, and probably well before that. But basically, be sure that you're picking up the new material here, and the main thing is to sort out which proteins are being utilized at each of these steps and how they relate to the cell cycle. Besides proteins, we're also keeping track of the chromosomes as well as the mitotic spindle, in other words, the cytoskeleton. Also, as a reminder, M phase is mitosis, those 5 events when we focus on chromosome segregation and the nuclear division. And then the very last part, the 6th phase, is cytokinesis, which is the division of the cytoplasm. So let's go through those in order. So prophase, this is when the mitotic spindle begins to form, and this results from the microtubules as they are finding the microtubule associated proteins. These are activated by MCDKs as they phosphorylate and stabilize the MAPs, and then the microtubules grow from each of the centrosomes. At this point, the nuclear envelope is intact, which you can see here, and the chromosomes are condensing. And so as we talked about before, that has to do with condensins and also cohesins. Then we go on to prometaphase over here. In some books this is called late prophase, but your book likes prometaphase. And this is when the nuclear envelope disassociates or disassembles. And so you can see it's kind of like this foggy stuff in the background here. And the microtubules will capture the chromosomes. Your book talks a whole bunch about how this happens using proteins like kinetochores, and that's just not something we're going to focus on, though it's very interesting. The metaphase phase, which you see over here, this is when the chromosomes line up at the equator of the spindle. So that's right here along this line. So they're in a perfect row. And this is what we call the metaphase plate. Going on to the last 3 phases of M phase. So here we see anaphase, when the sister chromatids are pulled apart. This is due to the cohesin complexes dissolving, and basically that means that each chromatid from the sister chromatid pair is pulled towards one pole. Just FYI, this is my favorite phase. And it's when the chromosomes are this little V-shaped material. One important note for our part of understanding the cell cycle is that if there's a chromosome that's not attached to the microtubule, then the anaphase promoting complex APC is blocked, and this thereby blocks mitosis, and the cell would hang out for some transient amount of time here in M phase and potentially be able to recapture that chromosome. Though it sounds a little bit unlikely, I'm just going to say it. Okay, then these 2 events are going on simultaneously, telophase and cytokinesis, so that's why they're put together here. You usually see these both occurring at the same time in animal cells. And so the mitotic spindle is disassembling, so basically the mitotic spindle has reeled in a chromosome and shortened up back to the centrosome at each of the poles, and then the microtubule spindle disassembles. You can still see the centrosomes here, these little glowing yellow things. And then the nuclear envelope starts to reassemble. So you can see that fog starting to form again. Then cytokinesis, this is when the cell is cleaved into 2 by the contractile ring, right along here. It's also known as a cleavage furrow, which is very blurry and impossible to read here. But the main point is we'll call it a contractile ring, and this is made of actin. So this is another role for the cytoskeleton in the M phase. Then the contraction of the contractile ring is due to actin and myosin.